Hey guys, I've gotten a couple emails about kind of reading the gels and understanding how that works. Um, so I'm making this short video to help you, hopefully help you understand. I know I'm going to be repeating a couple things from the PowerPoint you already watched, but hopefully this kind of little 5-minute crash course will help be a little bit easier. Okay, so the first thing to remember is that regular alleles are fundamentally different than STR alleles. So regular alleles, like if we were doing like eye color, for example, sorry guys. Okay, so if we were doing eye color, remember you would have one allele for eye color from your mom, and you would have another allele for your eye color from dad. The sequences are going to be really similar, like the one from mom and dad both start with AGGAT. But then on mom's, right here, it goes into CCA, and on dad's, same place, it goes TTC, and then they go back to matching after that, TCGGGAT. So again, our sequences actually have some base pairs that are different between them. With our, so again, that's that difference right there. However, with our STR alleles, the sequences are a lot more similar. Like here, they're starting out with the same thing, AGATT, and then they have this little, our actual little STR, our little repeating part here, CAT. So on the one from mom, we have CAT, 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 CAT. And on dad's, we have the same repeating part, CAT, CAT, but instead of just repeating 4 times like it repeated on mom's, it's going to repeat 6 times. Okay, so CAT, 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 CAT. Okay, so the sequences are pretty much the same. It's just the number of times that this little guy is repeating is different. And that's the difference between those two. So how do we make a genotype out of that? So remember, normally with alleles, we have a dominant allele and a recessive allele. So like with eye color, with our mom and dad over here, this person would be heterozygous because they have 2 different alleles. So maybe they're like big A, little a. Okay, but with our STR alleles, there's not a dominant and a recessive because this isn't coding. This is not coding DNA. So one can't be dominant over the other. So instead of having a dominant and a recessive, we just record the number of repeats in each sequence. So, you know, on the chromosome from mom, they had CAT repeat 4 times. So that allele is a 4. And on the chromosome from dad, that sequence repeated 6 times. So that allele would be a 6. So this person's genotype is just going to be 4, 6. Don't add the 2 amounts together and make it 10 or something, right? Because 10 could be 7,3 or 5,5 or 8,2 or any of those things, okay? So don't add them together, don't combine them, don't count the number of nucleotides. You're just paying attention to the number of repeats. So again, this person's genotype is not going to be, yes, they are heterozygous, but it's not going to be big A, little a. It's going to be 4, 6. So how do we actually get this information, or how would we be able to genotype somebody if we're just looking at a gel? So the way a gel works, think of a gel, an electrophoresis gel, like a freeway during a zombie apocalypse. You know, in all the movies, there's always just like tons of cars and semis and accidents, just like abandoned cars littered all over the freeway. Okay, so a gel would be like that freeway, and the DNA in this example would be like a motorcycle and a semi. So if you had somebody on a motorcycle that was up here and they were trying to drive through the wreckage, a motorcycle is small. It's going to be able to get between all the cars and stuff, so they're going to be able to travel pretty far. Whereas if somebody was driving a semi and they were trying to drive through this wreckage, like they wouldn't get very far at all. They would have a really hard time driving through this stuff. So when we look at a gel, or a picture of a gel, are smaller pieces of DNA, like the 4 that came from our mom. That one traveled further through the gel than the 6 that came from our dad. So, sorry, real quick. So these wells up here, these little rectangles up here, that's where you put your different samples. So you would dump your sample right up here, and then you would turn the electricity on, and the electricity pulls it through the gel. And so again, our big pieces, like the 6, can't travel as far as our smaller pieces, like a motorcycle, like the 4. So the smaller they are, the further they run down the gel. Okay, so again, this person was a 4, 6, right? So you can see the 4 from mom, the 6 from dad. We can clearly see this person is heterozygous because they have 2 different bands on the gel. They have 2 different alleles. So what if we do a different example? Let's look at somebody else who would be heterozygous. Okay, so again, we put one person's DNA in here, and then we have another person's DNA here. So we see this person has 2 bands on the gel. That means they have 2 different alleles, so that means they're heterozygous. And again, it's not big A, little a, we're looking at the numbers. So this person, it looks like they have one band at 16, okay, and they have another band at 8. So this person's genotype would be 16, 8. And then let's do one more example of somebody that's homozygous. Okay, so we've added our third sample here. It runs through the gel. So you can see this person only has in their whole lane, from top to bottom, there's only one band in the entire lane. And since that band matches up with 10, that means this person, again, is homozygous. So instead of being like big A, or little a, this person would be 10, 10. Okay, so this person's genotype would be 10, 10. All right, so I hope that helps you learn how to read the gels and how to make a gel. So let's look at our crime example real fast. So you guys, again, in your reading, you learned that the FBI looks at like 20 different loci, STRs in all those places. We're not going to have you look at 20 different loci. So yeah, what you do need to look at is just these 2 gels, and you need the person, or you need the samples to match at both loci in order for that person to match the sample. So when you're reading these gels, I realize that we don't have numbers on these ladders like I did on the example. That's not really important. I'm not worried about you guys being able to determine the length or how big these fragments are from the ladder over here. What you need to look at is where these samples match at. So when we look at lane 1, we see one band that's close to the top, so that means it's a bigger piece of DNA, and there's no other little pieces, so this person would be homozygous. And so when we look at the rest of the gel, we want to say, do we see any other pieces that have just one big piece at the top? We see in 6, 8, and 9 that there's a big piece at the top, but there's also like these fainter pieces at the bottom. So because these ones have a top and a bottom, top and a bottom, top and a bottom, and this one just has a big one at the top, they wouldn't match, okay? So now so 1 doesn't match anything. All right, so then let's look at 2. 2 has a piece here and a piece here. Do they match anything else on the gel? Well, if you look right next to them at 3, they do match, right? So we have another, like these 2 pieces are the same size, and these 2 pieces are the same size. And then if we keep looking down the gel, oh, we see that 2 and 3 match 5 also, right? Those are all the same size. When we look at 4, that has 3 bands in it. We don't see any other samples with 3 bands. So 4 doesn't match anything. We already said that 5 matches 2 and 3. When we look at 6, we have a larger piece at the top and then a small piece that ran down close to the bottom. Again, that's pretty faint, but it is there. So when we look at 6, definitely doesn't match 7, but it does match 8 and 9, because remember they both have big pieces at the top and then faint pieces close to the bottom. Okay, so from loci 1, we can tell that 2, 3, and 5 maybe belong to the same person, and 6, 8, and 9 also may, and then those would belong to another person, to somebody different. But again, we can't just, say that based on one gel. We need to look at both gels to decide what matches who here. So this is a really terrible picture. I might fix that. Okay, I think I might upload a different, better picture. These gels are really actually kind of crummy looking, you guys. We don't, in real life, our gels look a lot nicer than this. These were, run in class, so not super high quality. So anyway, okay, I think I might upload a different picture of gel 2. But again, just like gel 1, you're going to look through, you're going to see which samples match each other, and then when you look at gel 2, which is the second loci, you want to see which samples match each other. And if samples match on both gels, so if 2, 3, and 5, whatever those belong to, if those also match at loci 2, then we would call those a match. And that person would definitely be responsible for those samples would have come from them. So, okay, I hope that helps answer your questions. I'm going to post my phone number too, so you guys can call. I think that might be easier. Anyway, so I hope this helps. Okay, bye guys.